it. Thank you.
Do we need a treasurer's report? I know Paul would like to do the treasurer's report.
I didn't look.
We have
We'll
more, we have
forego
more
the
than $5,000 in the bank.
Sounds good to me.
Okay.
Rounsaville. You got it. All right. It is my privilege to introduce to you Todd Rounsaville. He received his Master's of Science in Horticulture from North Carolina State University and his PhD in Biology from the University of Kentucky. There, he also served as the curator of native plants at the UK Arboretum. He has also conducted research work at the Juniper Level Botanic Gardens, the Scott Arboretum, and the Arnold Arboretum. Currently, Todd leads the U.S. National Arboretum Germplasm Program to improve the genetic and taxonomic resource base of woody plants in cultivation. He conducts research on genetic diversity, seed biology, and germplasm management via regeneration, storage, and gap analysis. He leads and collaborates on expeditions for domestic and international plant collection, germplasm evaluation trials, and plant conservation efforts, both in situ and ex situ. Todd has recently served as chairman of a technical review team of 40+ scholars who produced the 2023 USDA Plant Hardiness Zone Map. He has published numerous studies on a variety of flora, including Lindera, Magnolia, Sorbus, Euonymus, and Miscanthus. This evening, for the 1048th meeting of the Washington Botanical Society, we are fortunate to have Todd with us, and he will present a talk entitled Seed Physiology Drives Plant Conservation Strategies. Can everyone see the slides?
I can.
Excellent. Take it away.
Thank you. Hi, everyone. Thank you very much for the invitation to speak to you. Congratulations to Greg. I don't think he's tuned in, but on his new child. This talk today, the subject matter is really plant conservation, but I want to look at it through the lens of seed and how we're using seed, some of the unique challenges and advantages of the work we do with seed, and also the work that's going on across the national plant germplasm system, which we are part of. So I'll start there to provide a little bit of context for those of you not familiar. This is a map of all of the germplasm repositories in the USDA National Plant Germplasm System. What we now know is NPGS began in the late 19th century with the USDA's plant introduction program to bring in new germplasm, both agronomic and ornamental, to assist in plant evaluations, breeding projects, ornamental trials, and has evolved to what we now know, which are 20 gene banks spread across the country. Any plant you can think of, any genus has a curator assigned to it that's embedded at one of these sites. And they're more or less laid out in regions that are conducive for their particular crop. So in Ames, Iowa, they curate corn. Geneva, New York has apples and grapes and hemp now. Most of these are agronomic focused crops, but Columbus, Ohio does herbaceous ornamentals. Miami, Florida does tropical ornamentals, and our site in Washington, D.C. is the catch-all for temperate woody plants. Headquarters of NPGS is in Beltsville, Maryland, and Fort Collins, Colorado. I'll return to them several slides, but they serve as the designated backup repository for all sites to do both clonal and seed cryo backup there, long-term storage. So our site, the National Arboretum, serves as the official repository, and that was designated, I think, in the mid-'80s, when it was kind of realized that forest genetic resources are just as valuable as agronomic crops. It's hard to count an exact number, but we're assigned somewhere around 220 genera temperate plants from around the world. And we manage those as the living plants at the Arboretum, but also our seed bank, which has about 4,000 collections at -20. All of the 20 sites across the country effectively have the same mission. It looks different depending on the crop, but we all have the same charge, which is to be acquiring new germplasm, bringing it on site to maintain, ideally in perpetuity, and while it's on site, doing different types of research. You know, we do genotyping and phenotyping. We do a lot of the work I'll talk about tonight on seed storage and seed dormancy. And then all of our collections are supposed to be made freely available to the public. And so we have an active distribution program, which is international, and we send seed cuttings, plants, pollen, anything requesters are looking for around the world. That distribution component is important for us if you are not familiar with it or ever have a need for germplasm in the future for any site. This is a few slides of the GRIN global website, which is kind of our clearinghouse for research requests for germplasm. So you can search on botanical name, or if you're at the Arboretum and see an accession number, you can search by accession number, you can search by country, and filter as necessary. This is an example of Cornus, Florida, so we'll pull up a list of everything that we have as plants or seeds. GRIN will show a map for the origin of any germplasm we have in the collection, and that can be added to a cart, just like your shopping on Amazon to check out. This is all provided at no cost. We have a pretty diverse stakeholder base, really anyone that has use of woody plant germplasm. We've gotten a lot of requests, maybe up to 10% recently, of people that are looking for seed that are archaeologists or doing paleobotany work, and they don't need to grow the seed necessarily. They're just trying to ID material that's been excavated. But we send a lot to botanic gardens and like that, because like us at the National Arboretum, they tend to maintain things long-term, so we can use them as a site to go back to if we lose something. And they also maintain wonderful records. But U.S. nursery and landscape industry is a big stakeholder, as are breeders and pathologists, people doing phylogenetic research. We do our best if we don't have something to try to find it for our stakeholders. So moving a little bit more specific into seed, the natural evolution of seed and their strategies for moving genetics in space and time makes them very conducive for a lot of the types of conservation work that we do. So from a collecting standpoint, it's a regenerative resource. We don't need to collect a lot of seed to be able to amass a lot of genetic diversity. Of course, if we're doing work, whether it's international or even domestic, we can very easily transport seed safely. Seeds obviously have developed the ability to go into a quiescent state that we call dormancy until the conditions are favorable for germination. And that allows us to store most seeds quite well. So in this middle slide, there's probably as many genotypes as there are at all of the botanic gardens in the world. And we can capture a lot of genetic information, very small space, and at a comparatively lower cost than the effort that goes into caring for plants and space they require for decades. And finally, the distribution component. It's very hard for us to send cuttings or live plants internationally to get them inspected and get them to their destination safely in time. So seeds make that a much more easier task to complete and be able to share wild species in particular with stakeholders. There's a ton of diversity in land plants, and of course their seeds as well. Just looking at some of temperate North America, the differences between something like Castanea, American chestnut, or this euonymus, when thinking about dry, spiny fruits versus smaller, fleshy fruits. Even within a genus like Acer, the maples, this is silver maple, Acer saccharinum on top, which those seeds shed over the last month probably. They have zero ability to dry down and be stored. They germinate right away. Acer circinatum, the vine maple, on the other hand, stores quite well. Those seeds mature in fall and are very hard to germinate, requiring multiple temperature cyclings to get them to break dormancy for many months. And for us, trying to collect quantity to be able to gene bank, there's a lot of differences if you compare something like oak leaf hydrangea, where in one seed head we might get tens of thousands of seed. And pyralaria, that's buffalo nut. We tried to do some collecting of that this past fall, and we're lucky to find 1 or 2 fruits per plant. So there's a lot of differences here that create unique challenges, species by species. So for the rest of this talk, I just wanted to cover some of those things, some of the inherent difficulties of working with seeds, and also some of the interesting opportunities that we can apply to use them in a meaningful way. Starting with that acquisition phase and just processing seeds that I hinted at before. These slides are from a trip to Japan. We don't do a ton of international work, and it's getting harder because of regulations with access and benefit sharing. We're told that we really can't collect as foreigners in China anymore, which is where the National Arboretum has a major part of their core collections came from. But we still do a little bit of international work. We collaborate with other agencies that can share international material, but for the most part, we're focusing on domestic collecting. This is a look at the accessions that we received just in the last calendar year. Most seed activities happen all at once in about a 2 or 3 month period starting in September. So it's kind of a frantic window of going to collect or receiving seeds and getting them processed and banked in a timely fashion. So our staff can do maybe 1 or 2 trips during that window, but for the Arboretum level involved in these multi-site collaboratives, I think I saw Alan Whittemore on the call, and who has done a lot of work with NACPEC in the past, who is focused on China collecting. So those groups are helpful to plan trips together and do seed sharing, because not all partners go on every trip. In the last few years, we've started to set up a contract collector network of people that we can pay, generally retired botanists that want to be in the field and know plants very well, get very good data. This is probably our most productive avenue for getting in germplasm. And then through the National Plant Germplasm System, they've set up a relationship with BLM and the Seeds of Success program, who are collecting across the country. It is pretty focused on Western states, but all of that seed is made available to the 20 gene banks I mentioned. And for anything that falls within your site's assigned crops, you can request that. And that brings in maybe 10 to 80 new accessions a year for us. Most of our processing is at a pretty small to medium scale. We shoot for 2,000 to 5,000 seeds in our seed bank, and any extra above that, we send to Fort Collins for long-term backup. But just to give a sense of some of the tools we use in case these are ever useful, having a ton of different sieves of different sizes work very well with both dry and fleshy fruits. This is a cheap new piece of equipment we got from Amazon. It's just a flour sifter, but we have had really good luck with things like rhododendron, ericaceous plants, plethora that are dry. We can just put them on this and it kind of vibrates, cleans the seed nicely. In the middle is an air column, which does a lot of heavy lifting for us to do separation of seeds from other floral material. And both pre and post cleaning, we have an X-ray cabinet now that helps some crops more than others do some preliminary screening of filled seeds. Give us at least a proxy sense of starting viability. So once our seeds get cleaned and are ready for storage, I just wanted to cover some of the different avenues for that. You've likely heard of some of these terms like orthodox and recalcitrant seeds before that. These terms refer to seeds' ability to dry, be dry to quite low moisture contents. Orthodox encompasses most land plants. 80+% plants are orthodox, and this is what we like. These can be dried to 5 to 10% moisture content, and generally can be stored that way at -20 or cooler for a very long time. So cleaning and processing those is quite easy. On the other end of this continuum are the recalcitrant species. So these tend to be, there's a higher proportion of tropical plants that are recalcitrant. A lot of large seeded things cannot be dried to more than 20 to 40% moisture content. And they can survive that, but they really can't be frozen without nucleation and damaging cells. So we do have very large collections of oak and magnolia for things like those taxa. We really have to resort to cultivating living plants and accommodating them at our site and sharing them with other sites. And of course, this is a continuum, so these are not strict categories, but there are some plants that they're not quite orthodox or recalcitrant, and they've kind of been given the moniker intermediate. So that's salix at the top, willow. Some intermediate things are able to be dried down. They can be stored cold, but they just have very, very short shelf life. And at the bottom in the middle, that's American beech, which is something that we're interested in at the moment. So beech is a relative of oak and chestnut, but unlike those two, which have no ability to dry down, it seems like from studies on European and Japanese beech that they may be in the orthodox to intermediate range. You're probably all familiar with beech, one of the most prevalent trees in Eastern North America. Some very impressive individuals, both at the Arboretum and downtown DC. So it's an important crop. There's only one species of beech in North America, and it's currently afflicted by a couple fairly serious diseases, and it would be helpful to have a means by which we can do safeguarding and backup, ideally with seeds, in case these diseases are anything similar to other things we've seen, chestnut blight or other forest pathogens. So based on some of the work that has been published on the other beech species around the world, we're looking at using liquid nitrogen, which beech seeds seem to respond to favorably, and doing some work to compare how dry we can get them. We know that very small gains in moisture content, if you can get it 1% lower, it can, for each 1%, do something like double its lifespan in cold storage. So we have a very small 20-liter liquid nitrogen tank that's useful just for some of this experimentation. Ultimately, what we would do is is back this stuff up at Fort Collins. So the image on the left is from maybe a month ago. The first round of seeds came out of liquid nitrogen and are doing fine, which is really nice to see, especially as whole seeds. The tanks on the right are just part of what's at the Fort Collins site. They have these massive tanks that have liquid nitrogen on tap. And really the bottleneck for them is developing protocols for how to get these things in. So what we're hoping to do to contribute is to work out optimizing the protocols and then being able to go to Fort Collins with new seed and some of the information they need to make sure things go into their tanks in the best possible condition. The team out there is doing some work, many different projects, but this is chestnut, which is very closely related to beech, but cannot be stored as whole seeds. They can be stored if you go and surgically remove just the embryonic axis, and that's what's here on the left, being dried down with nitrogen gas. But it takes an incredible amount of time and effort to, they're very vulnerable once they're cut out of the seeds, so they have to be managed much more carefully. And when they come out of storage, they can't be sown like we did with beech seeds. They have to go through sterilization and tissue culture, and that's riskier, more challenging, more costly, more time consuming. So when we can do anything as whole seeds, it's certainly ideal. We work with woody plants, which tend to have much more complex and deeper dormancy than a lot of other crops, and often not a lot of information on exactly how to break dormancy and germinate plants, germinate seeds. So just a few of the foundational texts that are great points of reference. On the left is Mariana Nikolaeva's Russian botanist. Her first volume, and then Carol and Jerry Baskin have kind of followed up on her system. Woody plants are challenging, and there's a lot of different dormancy complexes for us to be able to grow things, but also for our stakeholders when we send seed, to ensure that they're able to grow things efficiently and not waste the seed we send is really important. So this dormancy breaking is one of our biggest avenues of research. So I just wanted to focus on one thing, which is Viburnum lanthanoides. This is a North American viburnum, which is pretty underutilized, I think, in the trade. It has really incredible fall color, but it's ornamental in many other aspects. It has nice flowers, nice fruit. It is tolerant of incredibly dense shade, often growing like under hemlock canopy. And seems to, be one of many things that will be affected by a warming climate. This used to be in Kentucky. We went to look for it this past fall and told it's since been extirpated from Kentucky. We did some work with seeds that were collected in New Hampshire. So one of the things we know about all viburnum is that they have a type of dormancy known as morphophysiological dormancy, which is a two-part combinational dormancy where the morphological component is due to the fact that they have very small underdeveloped embryos. So first, the embryos have to be stimulated to grow, and once they have gotten bigger, then you have to break physiological dormancy, which is usually done through different temperature cycles. On the left is a pretty comprehensive phylogeny of viburnum, and they all have this MPD type dormancy, but there's many different types of that dormancy. And these relate to both the temperatures and the sequence of temperatures that are required in order to promote seed germination. Viburnum lanthanoides, you won't be able to see it here, but it falls in this very bottom group, for which there have not been any studies done yet. So this was something we get a lot of requests for, and we thought it would be valuable to try to characterize the specific dormancy pattern of the species. Baskins, who I mentioned in the previous slide, have proposed what they call a move-along experiment, which is their suggestion when you don't know anything about how seeds germinate. And the design of this is effectively using incubators that are set up to mimic seasonal temperatures, winter, early spring, late spring, summer. The spring temperatures double as fall temperatures, so we have multiple reps at each of these temperatures as controls, and then 2 treatments, which one of which starts at the winter temperature and cycles through seasons, and the other starts at the summer temperature and likewise cycles through. These images of embryo growth at the bottom are from the so-called warm move-along experiment. So what we're seeing is that, right at about this 10 to 13 week period, these seeds cycle from the summer to the fall temperatures. And we get this incredibly rapid growth of embryos, which then leads to, it's scored here as germination, but it's actually germination of the radicle only. So the seeds pictured here on right, which the audience may not be able to see, I think are from the 25-degree C control. So they've been like that for 18 months now. They've never seen cold, and they cannot grow without cold. Cold being required to break the shoot dormancy. So what we found is that these have to be cycled multiple times. First warm to have the embryos grow and break dormancy of the roots. Second, temperature cycle to cold in order to have break dormancy in the shoots, and then finally warm again to have plants actually grow. So our follow-up experiment is kind of just started, but is pictured here. We are trying to look at the minimum time required at both warm and cold to break these different types of dormancy, so that when we have stakeholder requests for the seed, we can provide guidance on the minimum, the easiest protocols for them to be able to grow these out, whatever their purpose may be. Seed regeneration, this was pictured on the left here, was a assessment that I think was published last year on from the National Academies on the native plant seed supply. One of the major findings of that is just that the demand for native plant seeds for all of these different purposes, the demand far outweighs the supply. And what is really needed is a non-existent industry, which is seed farming, especially for woody plants. It's a challenge for herbaceous plants and for grasses, but for woody plants, they get much bigger, have longer periods of time to mature, produce seed. The outcrossing, it's very hard to do controlled seed regeneration. So we've been doing a little bit of that for our seed bank collections, but only for select species where it's conducive. One of which has been Fopagilla. We've been doing a lot of regeneration on all of the species. If you know this plant and have seen it in the landscape, it's probably Gardeniae, which is a tetraploid, major, the hexaploid, or their hybrid Fopagilla intermedia, which is pentaploid. Within the last few years, there was a new species published, Fopagilla milleri, which is a diploid. Tends to be smaller in stature, smaller leaves. The leaves are very much, they're strongly vertically oriented. And because these seem to be able to cross ploidy levels, there may be some novel uses for people to do more breeding with some of these diploid species. As soon as Fopagilla milleri was published, it was extremely threatened. It's only known in a few populations in kind of the Florida panhandle and surrounding areas. There is a grad student in Florida, Clayton Hale, who did an ecological survey of all of the known sites a few years ago. And what they found in wild populations is that these plants cannot produce seed anymore. There's an aphid that parasitizes capsules at a fairly, early stage of development, and it leads to seed abortion. So any persistence of these populations is completely due to clonal ramets just slowly spreading. These have explosive dehiscence, kind of like witch hazel, which is the family that they're in. So this slide is a normal capsule, and right here is a parasitized capsule. So there's really no hope of getting seed from the wild or having any genetic recombination. Or so in the last year or so, we've been in contact with some of the Florida state botanists to try to assist them with regeneration and management. They sent us a handful of 40 or 50 seeds last June. I don't know if they had treated them with insecticide or June is before the aphid gets in and leads to seed abortion, but even in June, the embryos were fully developed. These also have not quite as complex as viburnum, but they have a fairly complex dormancy that takes months of temperature cycling. Because of that, and because they were still a little bit premature, we excised embryos. They grow very well just on filter paper. And this helped us really ramp up growth to get seedlings in 2 months. And then a month or two ago, we had hardened off plants that are ready to either go back to Florida. They would be our priority if they want plants back. The rest will hold and try to do seed regeneration on. So these tents are at our research farm in Beltsville, what we call South Farm. We have about 18 of them now. And they're really conducive for things like shrubs that don't get huge. We can plant them in these tents. We introduce bumblebee colonies, and they take care of all of the work for us. So we've had Fothergilla in here. I think this tent in the foreground has gardeniae, and I think we harvested almost 4,000 seeds out of that last year, which is pretty substantial. So the next few years, we're hoping to do more with Milleri to really bulk that up, get it in the seed bank, and have material for breeders or for conservationists that want to do restoration efforts. Data deficiency is something we run into all the time. So this figure, the histogram at the bottom is looking at all woody plants in North America, so almost 1,200 species. And it's a plot of the number of institutions that reported growing them. And that's data from the Botanical Gardens Conservation International group that aggregates botanic garden plant data from around the world. So what this is showing, there are 4 species that are way overrepresented. They're at over 250 institutions. Red oak, bald cypress, sweet gum are the 3 I remember. The biggest bin here is actually 0. So there's 65 or so plants that are not reported at any botanic garden. And so if you look at breaking all of these into quartiles, 25% of all North American woody plants are at 0 to 5 sites, 25% are at 6 to 17. Just as one metric that's been reported, seed storage behavior, what we know about it kind of scales negatively with the amount that a plant is grown in cultivation. So I think that's kind of telling in that the easier it is to access some of this material. If it's in a botanic garden, we get more requests for it. Of course, this is international, but most of this stuff at the bottom are the rarer plants, and you might expect that some of the rare plants would have or should have had a lot more research done for things like seed storage behavior. So that's something we run into a lot. As one example of that, I don't know how many of you are familiar with cider oxalon. This used to be Bromeliaa. It's a pan-tropical genus. This species is probably the most cold-hardy, northernmost species. And as this often happens, the Arboretum had some of these plants, cut them down, dropped the seed off with us, said you can seed bank this because the plants are all gone. Just checking the literature, there's no reports at all on storage capability. We looked at the germination literature and it said, if you do soak in concentrated sulfuric acid and then put them in cold moist storage and cycle them, that you might get very low levels, like 20% germination after many months, which seemed very strange. Also strange is these are reported to be troops in the literature when they're actually berries. And so all of the literature on germination recycles the same information from like a 100-year-old report. And there's been no follow-up studies. So our first approach was, again, the move along experiment to see what's going on. Are they really that hard to germinate? And from what we tried, they really weren't at all. We saw germination at every temperature. The treatment that started cold stratification and then was moved to warm did the best. That's pretty typical. It's what we might expect for most temperate plants that are used to maturing in fall, lying dormant for the winter, and then growing in spring. So it's curious, all of the previous reports that these are very hard to grow. Growing them in a lab setting, in Petri dishes also gives us an opportunity to make additional observations that could be relevant. One of those observations was not a new one, but that these have what's known as hypogeal germination. So instead of having cotyledons that come above soil level, they kind of function as a below ground storage organ. That's kind of like an oak would. That's sometimes been characterized with species that grow in heavily disturbed or fire adapted landscapes where that gives them potentially a little bit more protection or survival in the case of a disturbance event. The other thing we noticed is this kind of pocket of tissue you can see here in the top left. It's kind of whitish, and it's been described in the literature as like a fatty tissue deposit. And no one's really speculated what it does or why it's there. There's no connection of this tissue to the actual endospermatic cotyledons or the seed itself. But there is a second, water gap here for that to absorb water. So you can't see this in the audience with this. What we found when we were first hydrating these seeds is what would be called seed mucilage. And so this is an adaptation that a lot of species that grow in arid kind of desert conditions have evolved. And those systems are precipitation limited. So on the rarer events when they do get rain, this tissue, I'm now assuming, when it's hydrated, it produces this mucilage. If you've ever eaten chia seeds, you kind of know what mucilage is. And it would, in theory, help to buffer water intake for the rest of the seed to have the resources it would need to get that first push of germination. Why is any of that important? In this particular case, Sideroxylon happens to be one of the 100 or 120 or so species that the Forest Service has been kind of modeling as part of their climate change atlas. So they're looking at kind of modeling the fundamental niche of a bunch of native plants based on different climate change scenarios, taking into account, not just variables in terms of edaphic conditions or climate, but also some of these biological aspects of plants, including their seeds and how they disperse, how seedlings establish, and just adding data to studies like this, I think, can be meaningful for some some of this ecological type of modeling efforts. I'm gonna wrap up with an example of collaborative efforts, which is, really indispensable for plant conservation in situ or ex situ. We've been really fortunate to have the Botanic Gardens Conservation International Group to work with on a number of projects. They've started to they've started this global conservation consortia for select groups, including Magnolia, where partners from around the world are contributing data on their collections, but also research studies that collectively help all of us manage our collections better. The National Arboretum has been working on this particular species for well over a decade now. Magnolia asheyi is the rarest magnolia in North America, restricted just to the panhandle of Florida, and is kind of like big leaf magnolia, Magnolia macrophylla, but everything is a bit smaller. It's a smaller plant stature. The leaves and flowers are humongous, but a little bit smaller than macrophylla. And it's threatened by a lot of things down there, including fire, poaching, development in particular. Based on this gap analysis that BGCI led, there's in US and Canada less than about maybe 40 plants total. Magnolia is one of those species or one of those genera that are recalcitrant, so we can't bank them as seed. We have to, for the most part, use living plants as a conservation strategy. And the Arboretum, about maybe 7 or 8 years ago, initiated a genetic study to try to assess within and among population genetic diversity. They sampled all of the known wild major populations, but they also requested samples from all of the known materials in cultivation to be able to compare them, especially for those that didn't have provenance information to potentially place them. And one of the major findings of that study was that there was a particular site listed here as Holmes Creek that wasn't represented in ex situ conservation collections at all. And here's another figure from their gap analyses. This is the Holmes Creek central population circled in the middle here. We're seeing that there is pretty good coverage. The kind of filled in circles show where material exists in ex situ collection. So good coverage, but it's a little bit biased, heavy on the eastern populations. Some populations missing altogether. Because we need to grow these as plants and the resources and space to do that will always be limited. An interesting question that we sometimes think about but are usually struggle to answer is how many plants do we really need to capture a good complement of allelic diversity. And because we had produced a lot of the data, those were used in a follow-up meta study by Sean Hoban's group. This is Magnolia asheyi data, again circled here. So based on the survey of material in ex situ collections, they found that for this species, a little over 40% of low frequency alleles were captured. In some of their modeling efforts, and they're looking at across genera at oaks and cycads and magnolias, that for Magnolia asheyi, at the threshold date set of 95% capture of allelic diversity, that it would take at least 63 plants, provided random sampling across all populations. So that came out in 2020, and our follow-up work the past few years was effectively to try to fulfill that. This is Kevin Conrad pictured on the left. We went back in 2022 to sample all of these populations as randomly as possible, grow out seedlings where we've done our best to collect data and track all of these, at the maternal and individual level, and now we have way more plants than we need, maybe 400 now that we can hopefully distribute to Botanic Garden collaborators mostly in the US, but potentially internationally, to have a backup collection that we could go back to, or that Florida State botanists could go back to as a seed orchard, or to take cuttings from if any of these populations fail. And, you know, Kevin is one example of someone who's seen these populations for over a decade now. And some of them that he took me to when he first went had many dozens of plants. And one in particular had one tree left. So it is a bit of a sad story. So hopefully this contributes in some minor way. But I'll close there. Thank you for your attention. And if there are any questions, I'd be glad to answer. Yeah.
How do you determine how long these seeds are at their optimum viability? Do you mark them when you collect them, and that they're like 7 eons, or do they lose their viability at some point as in storage?
Yeah. So the question was, how do we know that seeds are still good and that they don't eventually die, which they will. Everything has a shelf life. I had some slides on that and took them out, but look, we try to test viability every 10 years if we're lucky. We've got thousands of accessions. If you were to do a better job, and as some people have, and plot viability over time, you get a very kind of sigmoidal curve where viability drops pretty rapidly. So if you're testing on a 10-year cycle, they could look good, and then in 10 years, they'd be all gone. And so what the group in Fort Collins is now doing, they were looking for a way to have a better assessment of that. And they've moved into looking at RNA degradation, which is linear. So they can look at the rate of RNA degradation over time and be able to determine kind of the drop-off point where when you reach, they have what's called a RIN number, which is the metric they assign. And when a seed collection gets to that level, they know from viability testing that it's about to collapse. And that's when they would do a backup regeneration or recollection. But every species is different, and some might last 5 years, and others, I mean, there's some of those seeds experiments you've heard about where it's been a century, you're still fine.
When you're growing out plants, either because the storage life is about over, or because, for instance, there's an insect that causes about 100% abortion out in the field, what do you do to try and maintain genetic diversity in a particular population that you are growing out for seed collection?
That's a very good question. Most of our colleagues at other gene banks that are working on corn and cotton, the breeding systems are very different to begin with, but they have the space to be able to grow out many, many genotypes and try to capture a pretty good complement of genetic diversity. Whereas for us, the seed we start with is all we have. We can't get any more genetic diversity if we're just doing regeneration at our site. So unlike most gene banks, I think in the National Plant Germplasm System, we know it's not ideal to regenerate woody plants for that reason, among others. So we do note that in our records. We note how many genotypes went into a regenerated sample. And for some things that is, better than nothing, or it can be very useful. But I would say, unlike most other sites, if we lose viability of our sample or it's in decline, our preferred option is actually to go recollect from the wild, to do it ourselves or to send someone else to bring in fresh material that has plant populations are still evolving in the wild, not in our seed bank. So never thinking that we can collect something once and it's all we might.
What kind of population are you looking at in terms of, I'm sorry, let me rephrase that. How many different genotypes, if you're growing something out, do you consider ideal?
If we're growing something out for regeneration,
Let's say it's not viable. It doesn't provide seeds in the wild because of some insect pressure or whatever. How many different individual genotypes do you consider to be the minimum you like to have?
I don't know if we put it, that's a great question. I don't know if we put a number on it. For us, if we're going to make a first collection in the wild, we try to randomly sample, within, a relatively contiguous population. I would say generally 10 to 20 maternal lines at a minimum. Some of that work we did with Magnolia ashii, there would be sites where there was only 3 to 10 plants, and maybe 1 or 2 of them would have seed, and maybe 1 or 2 of those would only have 3 pods, and then we try to collect ethically. So we either can't collect because there aren't enough, or we'll just get a couple seeds. So the ideal situation would be, probably 20 maternal lines at a minimum, but the material we work with is so diverse, it can be case by case. I think our best collection in the seed bank is oak leaf hydrangea, which is collected from almost every county where it's known across the Southeast, and in a small packet of those seeds, which were collected at the maternal level. So we may have hundreds of maternal lines with 50,000 seeds of each because they're so small and easy to collect. Question from the audience.
How much trouble do you have with self-incompatibility? Different sessions. You grow out one plant and is it going to set seed that's viable?
Yeah, a lot of what we work with are self-incompatible. So we try not to, at the moment, we haven't had a need to cross between, if we had a Georgia collection and an Alabama collection, we wouldn't cross between those. But we would have multiple lines that had come out of the wild, so we would cross.
You try to grow up multiple individuals from each population.
Yeah, and maybe have 10 to 20 unique genotypes contribute to a regeneration seed bulk effort.
So my question is, excuse me, are you collecting for just conservation purposes or also to introduce some of these to like the nursery trade?
I think if I were to ask our administrators, they would say that the U.S. nursery landscape industry is our biggest and most important stakeholder. And that's probably true. And we send them a ton of material. My personal interest is more, wild species and ecology. So a lot of what we're doing research-wise, kind of like, breaking dormancy in seeds, it's very relevant if you're a plant breeder and you need to grow something, but it's also very useful for the ecologist who's understanding population dynamics.
But I mean, as a homeowner, it'd be nice to have some of these native plants to, in the nurseries too.
Yeah.
So I'm just.
That's probably our biggest requester, looking at both at the breeders in academia, but also the bigger nurseries and other industry level breeders. We send them in botanic gardens more, send them more material than the other groups for sure.
No, I was just curious.
So are you keeping a database that keeps track of longevity and, and self-incompatibility kinds of problems that's publicly available?
The GRIN database where you could request material, some of the information you're asking about is publicly available, but a lot of it is tracked and is available on request. So, for instance, there's a viability component of the kind of behind the scenes website that staff have access to, or we're putting in viability numbers over time. I think I even saw Melanie Shorey on the line, who handles the entire taxonomy module, among other things. And I don't know how well fleshed out some of the sexual systems are in there, but usually if we're thinking about doing an increase, I'll just go to the literature, because I don't, just to confirm if something will work or not. I don't know it.
I don't keep any data on breeding systems in the taxonomy database.
Thank you.
Has there been much success with those gel coated cuttings of buds, like artificial seeds?
Is that for cryopreservation?
Yeah.
Yeah, I think the encapsulation, they maybe call it, where people are, stripping down embryos and then encapsulating them. But also, I think, as you said, with shoot axes,
Yeah.
you're doing encapsulation prior to freezing, and it's an extra step. So some of the people I know that are doing that work, if they can get them to survive without it, it's a preference. But sometimes it doesn't work, and it's an added layer of difficulty. But if that's what's necessary, then they definitely do that, like the sodium alginate beads.
Yeah. But that's a separate department from what you do. You're in seeds, and so there's a whole other germplasm department dedicated to cuttings, or?
In the Fort Collins site, there's a very big, seed group, and there's also the clonal group. Yeah, so the clonal group are doing more of that kind of work.
How are you keeping things from crossing with each other if you're growing up a couple different genotypes of some tree?
Yeah, I don't know if you could tell from that picture,
It
but
goes
there's
into the
those tents.
Yeah.
I don't think they're full, I think bees are pretty clever, and they may get out, but
How
We-
tall are those tents?
12 feet,
12 feet?
they're 12
They
feet.
get things that are flowering, trees that are flowering at 12 feet.
We're mostly doing shrubs, and
Shrubs.
it's just not practical to do trees in those, fortunately. That's another reason why we have to recollect.
Good.
There's a question on this.
Yeah, no, hi, it's Steve Lonker here. So have you guys tried the use of Bayesian models for determining the timing intervals for testing viability?
Say that again, Steve?
Okay, oh, sure. Have you guys tried Bayesian models, Bayesian statistical models for determining the intervals for testing viability?
No, I don't know if that's been done by any group. I have not seen that in the literature, but if you have any sources, I'd be very interested.
No, just curious, you're using that, yeah.
There's a question from Gene. Does the NPGS contribute to exchange seed banks outside the US? For example, the Millennium Seed Bank or Svalbard. The Fort Collins group definitely does back up its Svalbard for select crops. I don't know how they make those decisions, but I know they, on a fairly regular basis, the seed curator, Fort Collins, brings shipments to Svalbard. I don't know about Millennium Seed Bank, if we've done any exchanges with them or not. Melanie, feel free to chime in if you, oh, she did, yeah. Thank you.
I don't think we send anything to Millennium Seed Bank. We don't have a code for that. We definitely have a code for material that's sent to Svalbard.
We've been told recently that it's, for many countries, it's easier for someone to get an international user to get material from us in the US than from their own country, which is unfortunate, to say the least. But I guess it speaks to the efficiency of GRIN and NPGS that they can do it better than the host country.
What's the budget of your agency?
Melanie works at, I hate to put her on the spot, she works at headquarters, and I think
Yeah.
Melanie's lab, has historically helped significantly with funding a lot of those phyto.
So we pay for phytosanitary certificates for certain of the locations that send out material, and we pay shipping for some of them as well. And we've lost a scientist position because we don't have enough discretionary funding to rehire one of our scientists because we spend so much money sending seeds out.
You still interact with the Vavilov Institute, or is that kind of cut off?
Nothing is going to Russia or Iran or Cuba or China. There are political issues, and now there's a international agreement on material transfers that the lawyers have been churning through of how to have that implemented at the right time. So there are always new issues with trying to send material. And when we get material sent back by customs in various countries where either the requester hasn't gotten the right import paperwork, or they've changed the rules between when we inspected and sent the material and when the customs agents have received it. And when we get things back, it just has to be thrown out. We don't put material back into the collections after it's shipped out.
I have another question about genetics. In terms of collections, a lot of times natural areas, especially in the East, are quite fragmented. How do you handle the issue of cross-pollination, especially when a species is wind-pollinated, cross-pollination with plants in wild areas and then plants in gardens or domesticated areas? And especially, I'm wondering, there are a lot of species that they're cultivars for. So the genetics are identical for those cultivars in many cases because they're cloned. How do you handle the collection so that there is not an issue of not really having wild seeds?
Right. So with cultivars, if we grow them at the Arboretum, we wouldn't, collect seed except in rare circumstances, and we would send them clonally. I think to your main question about genetic pollution, I don't know what to say aside from we do the best we can to try to mitigate that type of pollution. It's pretty infrequent that pollen can travel significant distances. But it's pretty rare that we're collecting anywhere near development, at least the work we've been doing lately. We're usually in the middle of nowhere. And I don't know if we can ever be fully certain that even some of the plants there didn't escape, and we just assume that some of those individuals are naturally occurring, but it was just a long distance dispersal event. And we do our best to collect extensive data and to make all of that it's all publicly available in BRIN, but to be explicit about how many individuals we collect from, all of the GIS data, the proximity to any developed area is all very readily available to a requester. And, it's up to them, depending on their intended usage, to use their best judgment. But it's an important point, and there's only so much I think we can probably do. We're not genetically testing most of our collections, unless it's part of a research project.
Well, are there any other questions for the speaker? If not, we thank Todd very much for this very thought-provoking presentation, and we will see you all this time next month.
Thank you all.